Dominant epistasis. That is the second type of epistasis. The presence of single dominant allele at one locus masks the expression of alleles at another locus. So here we have two genes. One gene is acting as an epistatic gene and the other gene is acting as a hypostatic gene. So if dominant allele is present on an epistatic gene, then it masks the expression of hypostatic gene. So if single dominant allele is present at one gene, it masks or hides the expression of another gene. So its example is the fruit color in summer squash. So we have three colors: white, yellow, green. So here are the parents that are true breed. The white parent is crossed with green. F1 generation is white, that is indicating that white is dominant over green. In F2 generation, we have four conditions, four cases. And according to Mendel, the ratio should be 9:3:1. So 9 offsprings should have genotype A capital dash B capital dash. Again, dash is indicating that the homologue of A might be a dominant copy or a recessive copy, but the phenotype remains same. Similarly, the homologue of B might be a dominant copy or a recessive copy, but the phenotype remains the same because dominant alleles are present at both locus. So we have three cases with A capital dash small b, then three cases small a, capital B dash, and one case in homozygous recessive condition at both gene pairs. The first two cases responsible for white phenotype. Then small a, capital B dash is responsible for yellow color, while small b is responsible for green color. So that is a dominant epistasis, and it is indicating that if dominant allele is present at one locus, it hides the expression of another locus. So here A gene is acting as an epistatic gene, while B gene is a hypostatic gene. So if dominant copy of A gene is present, it masks the expression of B gene. So in first two cases, you can see that dominant copy of A gene is present. That's why it masks the expression of B gene at second locus and the phenotype is white. In case of third condition, A gene is present in recessive condition. So dominant copy is missing. So if dominant copy is missing, so obviously in that case, B gene can express itself. So when A gene is present in homozygous recessive condition and dominant copy of B gene is present, that is responsible for yellow phenotype. And in last case, A gene is present in homozygous recessive condition and B gene is also present in homozygous recessive condition. So here homozygous recessive B is responsible for green color. So B gene can express itself only if dominant copy of A gene is absent. So if dominant copy of A gene is absent, in that case, dominant B allele responsible for yellow color and recessive B allele responsible for green color. So the ratio for dominant epistasis is 12 white, 3 yellow, 1 green. Next is complementary epistasis. Two genes complement each other such that two dominant alleles are present at each locus. So in complementary epistasis, if two dominant alleles are present at both locus, then they modify the phenotype. For example, flower color in P. So we have two colors in P, purple and white flower. So here are the parents. So purple parent is crossed with white. Both are true breed. F1 generation is purple and F2 generation are again with 4 cases with a ratio of 9:3:1. So first case A capital dash B capital dash, phenotype is purple. So that is a complementary epistasis in which both genes complement each other. Both genes modify the phenotype. So here both genes have a dominant allele. That's why the phenotype is modified and it is responsible for purple color. The second case with dominant copy at first gene, but second gene is present in homozygous recessive condition. So the phenotype is white. Similarly, in the third case, when A gene is present in recessive condition and B dominant allele is present, but the phenotype remains white. And similarly, in the last case, when both genes are present in homozygous recessive condition, the phenotype remains same. So here, the ratio is 9 purple and 7 white. So that is a complementary epistasis in which if dominant alleles are present at both locus, then they can modify the effect. So here it is, the picture is showing the mechanism of action of the gene A and gene B in complementary epistasis. So we have a precursor molecule and that is a colorless molecule. So if gene A is present with its dominant copy, that will convert the precursor into intermediate product and that is also colorless. After the formation of intermediate product, gene B came with its dominant copy and that convert the intermediate into final product and that final product gives the purple color. So both genes should contain dominant alleles for the formation of purple color. If dominant copy is missing from any step, then final product is not formed and that's why the phenotype should be white. Another type of epistasis is polymeric gene interaction or polymeric epistasis. It is defined as two dominant alleles have similar effect when they are present separately, but produced enhanced effect when came in combination. So we have an example that is a fruit shape in summer squash. We have three shapes in summer squash: disc shape, sphere shape, long shape. So here are the parents. Disc parent is crossed with long parent. Both are true breed. F1 generation is disc. F2 generation is again with 4 cases and the ratio should be 9:3:1 according to Mendelism. So we have a first case with the A dominant dash, B dominant dash and its phenotype is disc. Second, A dominant dash and B is present in recessive condition. Phenotype is sphere. Third case, A is present in recessive condition while B dominant allele is present. So that is again responsible for sphere shape. And in last case, when both genes are present in homozygous recessive condition and dominant alleles are absent at both locus, that is responsible for long shape. So that is a polymeric gene interaction. That is saying that if dominant allele are present at any locus, then they are responsible for same phenotype. It means if A dominant or B dominant alleles are present separately, they are responsible for same phenotype. That is the sphere shape. But if both dominant alleles of both genes come together, came in combination, then they are responsible for disc shape, means they are modifying the effect of each other. That's why the shape is disc. And in last case, when dominant alleles are absent at both locus, then it is responsible for long shape. So the ratio for polymeric gene interaction is 9 disc, 6 sphere, 1 long. So again, polymeric gene interaction is that case in which if dominant allele of the both genes are present separately, they are responsible for same phenotype. But if they came together, they modify the effect of each other and they responsible for different phenotype. So that's all about the epistasis.